This is the problem, correlated with the presence of infectious virus. The absence of genome is the proof of the absence of infectious virus, because without genome, the virus cannot be replicated. But you may, with the right control, because there is a lot of problem of inhibition and so on. And the absence of viral genome may not give any information about the global fecal pollution or the presence of other viruses. We are at the other extreme. When you have a fecal pollution indicator, it's a very global information. When you go to detection of a norovirus, you are looking for a norovirus. And the only information you get is there is no norovirus in my sample with my primer I use. Because we saw also that norovirus has a high variability genetically. So all the time you have to check your primers to be sure that you are looking for the right virus. So it's very, very specific. We are on the other range. And we cannot use molecular tool to estimate the treatment because you see that there is a large underestimation of the impact of treatment. Okay. So let's put molecular biology on our graph. If we have high fecal pollution, no need to go. But low fecal pollution, perhaps we can make some PCR. But now, if you have a negative results, okay, it works. We have no viral pollution. But when you have a positive results, you again don't know where you are. If you have a high probability of infectivity or low probability of infectivity. Okay. And now, let's try to interpret the result I showed you yesterday. How can we say, what happened, what are these 75% positive? Are they infectious virus or not? You don't know. If you don't know, you have to say, okay, it is infectious, because you have to make precaution, of course. And it's the same thing for the other positive results. And this is the main problem. If you have time at the end, I will show you some thinking we made at the French Agency for Food Security some years ago. Okay, at the end, if you have time. So what should be done? We should use the standardized method, of course, to evaluate the presence of genome prevalence. We have to quantify viral genome to evaluate viral pollution in different matrices to have an idea what is the level. Try to make difference between infectious, try to make links between the detection of genome and epidemics. To try to see the links and better understand mechanism of inactivation to know when viral genome is okay for analysis and when it is not. But we should not forget that we have other tools, like fecal in the bacteriophages also. Just few words about this. Bacteriophages are the most abundant biological entities. We have different bacteriophages, mainly 79% are viruses of bacteriophages are viruses of bacteria, of course. They are mainly a head and a tail. For in 90–70% of the species know yet there are these structures, but some of them have a lot of different other structures for 3%. Some bacteriophages are also present in human stools. And somatic coliphages are very frequently found in human stools, from 20 to 90%, at very high concentration. F-specific RNA phages also are found sometimes at lower concentration, and B fragilis phages, Bacteroides fragilis phages, are also found in human and animal stools. Of course, then, you find them in wastewater. We made some European project some years ago with Colombia, Argentina, France, Spain, and we check what was the level of F-specific, of bacteriophages here, somatic coliphages, F-specific RNA phages, B. fragilis phages, compared to fecal coliforms, fecal enterococci, and some spores. And you see that we can find, this is in log, so you can find 10 to 6 fecal coliforms, where you have 10 to 6 also somatic coliphages, a little bit less F-specific and a little bit less B. fragilis. They can be found all over the world, more or less. The advantage of this is that bacteriophages then are viruses, and they behave like viruses. That's the difference of bacteria. Somatic coliphages are bacteriophages that infect E. coli by a receptor on the membrane of the bacteria. Mainly, they belong to 4 different families, because it is important in terms of structures. 3 of these family, Myoviridae, Cephuviridae, Podobiridae, are head-tailed phages. And you have also Microviridae, which is a family of bacteriophages which have a structure more or less the same as enteric pathogenic viruses. But there is a very difference. You detect all of this when you are looking for somatic bacteriophages. F-specific phages, you have DNA phages and RNA phages. When you are looking at F-specific RNA phages, meaning that the phage has an RNA genome, and this was as a DNA genome, this phage infects E. coli by the F. pili structures, and you see that we have a similar structure that enteric viruses. We have here cubic capsid protein with inside an RNA, like norovirus, for example. And finally, B. fragilis phages belongs to one family, which is head-tailed bacteriophages. These phages usually don't replicate in the environment because main 2 parameters. One is the abundance of the host, which is low in environment, and the other is the metabolism of the bacteria. So, phages are fecal indicators, okay, which are not pathogenic, of course, for human. They may not replicate in environment, easily detectable with low-cost method, and their infectious character can be easily checked with very simple method. With the plaque-forming units, you just put on a plate, and you get some plaque-forming units. We have standardized method available for several years. And some phage, I just show you, have very similar structures as enteric viruses. They have sometimes a similar survival, and sometimes not, of course. Okay. And to finish with phages, I will not say a lot about this, but it's also interesting. Some of them may be also tools to discriminate the origin of fecal pollution. This is another European project we were involved. We were looking about 20 different parameters able to discriminate the fecal origin of pollution. 20 different parameters plus 18 associate parameters, meaning that we use not only 1 but 2. And we analyzed 100 samples from known origin, and what we know is that the best tool at this time was to look about somatic coliphages associated with B. fragilis phages, which was a special one. Okay, I will not go to details, but you see here, if you look concentration of somatic coliphages and put here the ratio between somatic coliphages and B. fragilis phages, you have here all the samples coming from animals, and here all the samples coming from human. This means that you can differentiate very well both of the origin. The genotyping of a specific RNA phages was also interesting. Among all these parameters, these two parameters were the most interesting. Okay, now again, when are bacteriophages interesting? Again, when we have low fecal pollution defined by bacteria. And in this case, perhaps we have to choose the right bacteriophages. And then if you have positive, in my opinion, there is also a high risk to get infectious enteric viruses. If we have low, perhaps you have lower risk, but not in all situations. It's more or less difficult. Okay. So this is the one part of the talk. Okay, let's go for the detection. Now, the survival. To define survival of a virus, we need a virus which is able to be cultivated. So we need cell line. So you have to choose a model. I will not go to different details, but you can, what is usually made is use a virus from the same family which is cultivable. For norovirus, for example, you can use the murine norovirus, which may be cultivated cell line. And then you can follow the survival of this virus. You can use same structured viruses. So perhaps you can use F-specific RNA phages to estimate the survival, or choose the most resistant. This is the best choice in my opinion, choose the most resistant, so you're very sure that for all the others your treatment will be at least this. But the information is not easy to get. Which is the most resistant virus? We don't know, actually. This is why we make some effort to put together all the literature information to try to say which one is the most resistant. So we make a lot of literature reviews, and I just want to show you 3 examples, if I have time. First one, I feel very confident now to say that UV, if you use UV, MS2 phage, which is a F-specific RNA phages, is a very good choice. I will show you the results. If you use a heat treatment, which is not very easy for water, of course, but heat treatment, these are the most 2 parameters which define survival of virus in environment. It's sun and temperature. Heat, we have also some phages which are very resistant. And finally, I will open the discussion to show you some results which I may not explain at the present time. So first, this was done in a cost action, a European project. We just put all the information of literature. We select some papers, and use this paper, we put some meta-analysis of this. What appeared? This try to summarize on this graph. All the results are here. There are on these lines here are all the data for 14 publications, 64 log reduction from literature, and you see that all the data are here for F-specific MS2 phage. MS2 phage, you see here you have the UV dose and inactivation in log. So all the data are here for MS2. And these are all the data for all the other viruses, meaning murine calicivirus, all the data, phages. So this means that you see that we can very easily define that MS2 phage is the most resistant for UV treatment. Globally, we have 4 log decrease for 60 millijoule per centimeter square. So this is the classification we can make. Adenovirus feel a little bit better. Phages, MS2 phage. These both viruses are the most resistant to UV treatment. So if you want to check your UV treatment, use MS2 or adenovirus. And now we know, I will not go in details today, but we know why. Because UV impacted genome.